A groundbreaking study led by researchers at National Jewish Health has uncovered new insights into the role of macrophages in lung injury and repair. Macrophages are immune cells that play a critical role in lung health, especially during times of injury or disease. These cells help fight infections and also participate in the healing process, though they can sometimes contribute to lung fibrosis, an irreversible scarring of lung tissue. Historically, certain macrophages were thought to be primarily responsible for promoting fibrosis. However, this new research challenges that belief, revealing that the so-called pro-fibrotic macrophages may not be as closely linked to fibrosis as once thought. This discovery opens up new possibilities for how we might harness macrophages to encourage healthier lung repair, rather than allowing them to contribute to scarring. Macrophages are immune cells that reside in the lungs and protect the body from harmful invaders like bacteria, viruses, and fungi. They also play a crucial role in the body's response to lung injury, whether from infections, pollutants, or other diseases. Upon injury, macrophages are recruited in large numbers to the site, where they help initiate repair and, ideally, restore lung function. However, in some cases, instead of promoting healing, macrophages can contribute to the development of lung fibrosis, which leads to stiff, scarred lung tissue that impairs breathing. This study sought to understand why some macrophages lead to successful lung repair, while others promote fibrosis. Researchers focused on comparing macrophage activity in two different models of lung injury. One model was associated with healthy repair, while the other led to fibrosis. They wanted to identify the differences in the macrophage populations in each scenario. The team's initial findings revealed that certain populations of macrophages were recruited to the lungs in response to both types of injury. By analyzing the genes expressed by these macrophages at different time points during the healing process, the researchers found that macrophages expressing a gene called Gpnmb, historically described as pro-fibrotic, were present in both types of injury. This suggests that these macrophages may not be as crucial to fibrosis as previously believed. Further investigation showed that these Gpnmb-expressing macrophages were present in lung tissue samples from a variety of diseases, including asthma and COVID-19, which have vastly different recovery outcomes. This finding led researchers to reconsider what factors influence whether lung injury results in fibrosis or successful repair. The study also highlighted the role of the lung's microenvironment. The subtle changes in protein expression and macrophage survival in the fibrotic injury model were linked to activation of distinct pathways, which contributed to less effective healing. These findings suggest that the lung environment itself plays a critical role in determining whether macrophages promote fibrosis or healthy repair. With this new understanding of how macrophages behave in response to lung injury, researchers now aim to explore what exactly triggers these differences in macrophage behavior. Their ultimate goal is to determine if it's possible to redirect macrophages, particularly those linked to fibrosis, to instead contribute to healthy lung repair. Dr. William Jansen, one of the lead researchers, emphasized that the research marks an important step toward rebuilding injured lungs. By understanding how macrophages are programmed, scientists hope to develop strategies that could encourage healing and potentially prevent or reverse fibrosis in lung diseases. This research has the potential to revolutionize the treatment of chronic lung conditions and offers hope for those suffering from diseases that currently have few effective treatments. The future of lung repair may lie in the very cells that have been seen as the culprits in fibrosis all along. Stay tuned for more updates as this exciting research progresses and brings us closer to understanding how we can better heal damaged lungs.